So welcome, we're going to go ahead and get started. The recording has begun. We will be sharing the recording of both days of the session online on the Open Oximetry website. Sorry, there's a few technical issues here. Okay, great. So day one. Welcome to day one. The purpose of today is we're really going to try and put everything out on the table in terms of challenges, problems. This is going to be a discussion of the problems that have been coming up lately with pulse oximetry. This issue of performance, accuracy, potentially due to skin pigment is going to be a lot of the focus, but there are going to be other issues that we're going to want to cover. We'll go a little bit more through that agenda in a moment. Day two is going to be more discussion focused on solutions, trying to brainstorm, people putting forward ideas and thoughts. The focus is on how to modify the ISO standard. That is part of the reason why we're here today. And so those problem and solution discussions, think of it please through that lens. There are a million things that we probably could discuss with the expertise and experience in this room, but we do want to try and keep it focused to actionable next steps for the ISO standard. So just keep that in mind, please. Day three, the ISO and IC committees will be convening a closed meeting where they will be synthesizing the information discussed in these first 2 days and trying to work on revising that standard. For those of you who are part of that committee, the meeting will take place on campus here at UCSF, and we're happy to share more details, and we'll do that after the meetings today. So please find either myself or Fifi if you have any questions about that. As mentioned, there's a Google document which we will put in the chat right now on Zoom for those who want to follow. Apologies to those from certain institutions who don't have access to Google Documents. We will share a Word version of that later. But for now, that document's going to be open. Anybody can edit. So if you want to add to the conversation, you think something wasn't captured that was in the meeting, but we will be keeping minutes as we go, and we'll share those afterwards. But that document is open. That document also contains some of the discussion, a list of challenges, research priorities, issues that should be addressed from our last Open Oximetry meeting in December. So there's a list there in that same minutes document. The same minutes document also has the agenda for today. So if you're wondering what's coming up next or what to expect the rest of the day, everything's in one place. So we'll share that list. If anybody, again, doesn't have access to the Google Doc, needs something, please email either myself or Fifi, and we will get that to you today. The structure of each of the sessions is a little bit different. We have panels, presenters rather, speaking. There'll be a moderator. Everybody's going to be whoever's presenting will be sharing from their own computer. And we'll talk about some of those logistics, and the moderators will be doing very brief introductions just because of our tight timeline. So any speakers who want to elaborate on their introduction, anything that's relevant to why they're here today and what their talk is on, please elaborate. Don't hold back. That's okay. And then at the end of each day, we're going to have some open time, open forum. We sent an email out to Open Oximetry collaborators, anybody who wanted to have time to share. And so that will be open. So there are a few folks who have signed up. We may have a few others who trickle in, and then anybody who's participating who wants to share, that would be the time to do so. And then the final piece of each day will be a moderated discussion trying to synthesize all the points from that day. And again, another opportunity for general discussion. Just as a reminder for those who are joining through Zoom, everybody who's here in San Francisco, please do not join computer audio. That will avoid echo problems. And then everybody who's online, please stay on mute when you're not speaking. If you do want to speak, please do so by using the chat, and you can chat to us, or by raising your hand. For those of you in the room, here's the how to leave your computer audio. At the bottom left, click the caret next to the mute or unmute button. The bottom option is leave computer audio, just as a reminder. And then for those of you who have not used Zoom very much, good for you for avoiding that over the last 2 years. But here's the way to raise your hand. So reactions button in the bottom menu bar of your Zoom, you'll click on that, and then there's the raise hand. Don't forget to lower your hand after you've already said what you want to say, and the moderators will try and field those questions as best we can. If we can't get to it again, then please put it in the chat, and we'll try and bring it to the end of the day when we have the dedicated discussion time. One last logistical thing with Zoom. We've been shifting around the Zoom link. Apologies. Many of you who are logging in via Zoom have my personal Zoom login. So please look at your name and make sure that your name is you and not me. To the extent that you're able to do that, you can do that in the participant list. You'll click on the 3 dots next to your name and then rename yourself. Again, go to participants in the bottom menu, find yourself, which should be at the top, click on the 3 dots next to the right of your name, and then select rename, and you'll be able to put your title. For everyone, please adjust your affiliation and say where you're from, just so people know in the chat. And hopefully we'll get an opportunity over the course of the next 2 days where everybody will be able to introduce themselves at some point, so that we can everybody can know who's here. Before we move on, are there any logistical questions about Zoom audio or otherwise before we launch into things? Okay, great. So the reason that we're all here, just to frame the discussion a bit, really boils down to patient safety and issues of health equity for everybody. I think over the last couple of years, everybody in this room has had either a lot of experience with, or certainly become very familiar with a lot of the buzz, a lot of the research that's been coming out about oximetry performance. A goal for today is not to rehash all that, but really to try and think about actionable next steps about how we can improve patient safety and health equity. As mentioned before, a lot of the focus is the ISO standard revision, so please keep that in mind as we may be refocusing discussion. As the moderator, I will try to help do that, so please don't be offended if we kind of put some things in the parking lot so that we can keep conversation going in a productive direction. There's a lot that can be said. We, again, want to have a very open conversation, but we will need to have some bounds on what we can discuss in this next few days, because it's a finite period of time, and we do want to make some progress here. The emphasis is going to be on diversity and device testing and applicability. That's going to be a big part of the discussion. A lot of the challenges will be discussed today, and the morning tomorrow is really dedicated to that. Performance during challenging scenarios, including low perfusion, is one that has come up, but perhaps others is also going to be another issue that's very relevant to the standard. And then finally, improving communication about performance to end users, to clinicians. How can we leverage the standard to do a better job of doing that? That certainly has come up as a big issue over the last couple of years. We've heard a lot, as I'm sure you have, of I had no idea that is, my device might not be telling me what I think it is, or I shouldn't use it in this way. So how can we leverage the standard to improve communication around that issue? Also, not all devices are the same. And it's confusing to consumers. It's confusing to those who procure these devices, whether it's a Ministry of Health or an aid agency, to understand what the different classes of devices are, what a difference one certification means versus another. There's a lot of room that can be done with harmonization of regulatory, and it's possible that the standard could be used to try and drive some of that discussion. So I'm highlighting those as kind of 3 big objectives that we're hoping that we can accomplish over the next 2 days. Additionally, gathering thoughts from diverse stakeholders is a big goal for this as well. We have folks who will be joining in from different countries around the world, but also many different disciplines that weren't part of conversations up until now that we think have a lot to offer. And we're hoping to create an open space where people can share their thoughts and we can learn from each other. There's a lot of ways that we anticipate that will provide for an interesting discussion over the next few days. So please approach this with an open mind and introduce yourselves and meet some people from other disciplines and places. Lastly, pulse oximetry standard has the potential to have impact beyond pulse oximetry. There are a lot of devices that may have some of the same challenges that we're going to be discussing today. So please keep that in the back of your mind. What we discuss over the next 2 days and how that impacts the Pulse Ox standard may have implications for other standards. So finally, I will stop talking about why we're here, but I think that is sums it up a bit. Everybody may have slightly different perspectives, and please feel free to share those as we get going. Timeline-wise, up until now, I think most people are very familiar with this, highlighting over the last 40, 50 years, data that was suggesting there were inaccuracy problems due to skin color, for example, but really during COVID is when this was linked with health and healthcare disparities. It was also a time when we were seeing large-scale donations of devices that probably were not fit for clinical use, and it wasn't always clear why that was the case. So 2 distinct issues, but related to certainly improving the standard. In 2021, a lot of global initiatives began to respond to some of these problems. in this room are some of those entities, but we don't claim to represent all of them. And we know that there are many that are ongoing and have tried to invite as many to this conversation as we can. The FDA safety communications in 2021 and 2022, the FDA open public hearing in November of last year. And then since the open public hearing, the Open Oximetry Collaborators Group has had multiple meetings. There was one larger meeting in December and then several subgroup meetings to discuss more specific issues. And all of those have sort of helped form and shape the meeting agenda for the next couple of days. There's a lot that we think we know. I don't claim that this is an exhaustive list, but we know that, or at least we think we know that performance of many oximeters is impacted by the issue of skin color. Bias has now been correlated with health and healthcare disparities, bias due to race, that is. Common methods for describing skin color are flawed. Diversity and sample size requirements for pulse ox standards are clearly inadequate. The current standards do not address all known confounders, including some that are very relevant. Some clinicians and end users, again, don't understand or maybe have not had a recent refresher on the limitations of proper pulse oximeter use and interpretation. And then also, we know that more diverse stakeholder input is needed to address this problem adequately. What we don't know is the magnitude or the root causes of some of these performance problems, especially due to skin color. And we will dig deeper into that a little bit. How best to assess skin color, melanin, other tissue physioptic properties. We don't know which we should be accounting for and which perhaps not during the ISO, in the standard. The full relationship between skin color and pulse oximetry, we don't know that. How verification tests can be improved while balancing cost, the time, complexity with things that may translate into access issues or ethical challenges, which confounders to add to the standard, and many other things. Again, not an exhaustive list. I'm going to stop speaking there, and we're going to get ready to welcome some other speakers. But I just want to thank Fifi Nagusi from our team here at UCSF, who's really led the coordination of this meeting. She's done a phenomenal job doing that. Also, Patty Orozco and Zaina Tomalea from the CHESA team, Bob Kapadik and Sandy Weiniger from the ISO committee for also helping to coordinate this meeting, and many others, including all of the speakers from today, as well as some of the funders who enabled us to host this meeting, for those of you who are here with us in San Francisco. So with that, I'm going to go ahead and stop sharing my screen. And we're going to get ready to jump into the agenda. Dr. Ehi, are you online and available to begin your presentation?
I am. Can you hear me okay?
Okay, yeah, great. And if you have slides to share.
I'm begin doing so then. Give me a second.
Great. That sounds great. While you're doing that, I'm going to go ahead and introduce you. Dr. Odi Ehi is an associate clinical professor here at the University of California, San Francisco, pediatric anesthesiologist, part of our CHESA team and part of the Open Oximetry Collaborators team. She's also the vice chair for diversity, equity, and inclusion in the Department of Anesthesia at UCSF. And we're very lucky to have her joining us today to share some of her thoughts on this topic and to help us kick off this meeting.
Thank you so much for that introduction. I'm just gonna give me a second to get this up and running. I have to adjust here. Just let me know when you can see the entirety of my slides.
Perfect. Looks great.
Fantastic. Again, thank you all. I wanted to use this opportunity to kind of give you a brief overview of how pulse ox bias relates to structural racism, as well as the importance of applying a DEI lens to achieve health equity. My role is the vice chair for diversity, equity, inclusion within the Department of Anesthesia, as Mike had mentioned. I also wear a couple of hats within JEDI committees and DEI committees within California, as well as nationally within the Society of Pediatric Anesthesia. And to just disclose, I am a pediatric anesthesiologist in practice. But again, I wanted to really focus more on why do we need a DEI lens as we approach this topic of pulse oximetry and bias. So I think the first thing to do here is at least to define the different terms before we move forward. And then we can delve a little bit deeper into the use of these terminologies and how it's related to health disparities. So when looking at a general definition of race, it usually refers to a social concept used to describe a group of people who share physical characteristics such as skin color and facial features. And ethnicity is usually referred to as a social group that shares a common and distinctive culture, religion, language, or the like. Whereas skin color, a very rudimentary description of that would be a phenotypic expression depicted by melanin, which has two forms, eumelanin, which is the black brownish component, and then pheomelanin, which is the yellowish reddish component. But when defining skin color, it's really important to state that the size and number of these melanin particles can vary and are even more significant in human skin color than the proportion of these two types of melanin. Also, we know that some skin-related factors, such as keratin, influence skin color as well. I know Mike has already referred to all the physiological components, vasodilation, all these different physiologic pieces that lend itself to skin color. And we can really delve, so much more deeper into this, but we don't have the time. So we'll stick with this rudimentary definition and move forward with why we're speaking about this. So one of the ways that that skin color and race has been defined, actually, I think this article does a really good job of it, is to state that skin color is the primary physical criterion by which people have classified or have been classified into groups in the Western scientific tradition. This article actually goes on to say how skin color has been the primary characteristic used to assign people to different races for social reasons. So to further delve into this, there's actually a JAMA article that was placed out in 2021 that really focused on an updated guidance for reporting race and ethnicity in the medical and science journal. And when it defines race and ethnicity, it actually, more importantly, goes on to state what they are not. So race and ethnicity are social constructs without scientific or biological meaning. I think we have to be clear on this. And this article then goes on to acknowledge how the indistinct construct of racial and ethnic categories have actually been increasingly made aware in the area of medicine and science, but also acknowledges how concerns about the use of these terms in medical and health research, as well as in education and in practice, have been progressively recognized. And you can see within the past few years, there's just been so many articles and so many studies that have come out in relation to all of this. So one of the things I really want to highlight here in being more specific about race and ethnicity is that we know that historically, although inappropriately, race has been considered by some to be a biological construct. And as a result, some older research has actually been known to characterize race as having biological significance, which has led to some harm. And we can go a little bit more in detail with some of the examples that you will see later. But we have to realize that these antiquated and these erroneous ideas of using race as a biological construct as a premise can further perpetuate health disparities through structural racism. So how do we report on race and ethnicity? Well, first of all, the reporting of race and ethnicity should not be considered in isolation. We should actually be thinking about accompanying it with reporting socio-demographic information, social determinants, including concerns about racism itself, disparities, inequities, as well as intersectionalities of race and ethnicities with other factors. And this actually brings me to what are some even myths about color. So how many times have you heard that individuals with skin of color don't need to wear sunscreen or are safe from skin cancer? We know that that's not true. And even once risk for sunburn is affected by skin type, the medications that they take, their length in the sun, we know that we have to realize that it is a myth that anyone is immune to sunburn. So really kind of taking away that one person is different, all just because of their skin tone, I think is really what I'm trying to push forward here. And also there's this myth that people of color don't get skin cancer, which actually is harmful. We know that because of this belief, skin cancer has actually been oftentimes diagnosed much later for people with darker skin. So as we move forward to bridge the gap of health equity with relation to skin color, we have to examine structural racism. So let's get into that. What do I mean by structural racism? Well, the Associated Press defines it as structures, policies, practices, and norms resulting in different access to the goods, services, opportunities of society by race. In other words, it's how major systems like the economy, politics, education, criminal justice, health, can perpetuate unfair advantages. Also, the Associated Press actually advises not to shorten this term structural racism to the term racism to avoid confusion with other definitions. So when we speak of structural racism, we'll keep it holistic in that component. So now let's get into why this is relevant for the Pulse Oximetry project. Well, there's evidence that minoritized populations are much less likely than their white counterparts to be included in studies on environmentally related diseases. Even those disproportionately that affect minoritized communities, they are less likely still to be included in that. And this article goes on to say how there's been a failure to create more racially diverse research cohorts, which could exacerbate health disparities, and according to some experts, actually does. And here's an example. We know now that Black and Latinx groups make up 30% of the US population, but account for just 6% of all participants in federally funded clinical trials. And there's several possible reasons why minorities are underrepresented in biomedical research studies as well, some of which include cultural and linguistic differences, as well as financial and time constraints. For example, a subject not having time or money to travel to a study site. And this can thus potentially lead to less participation from minoritized groups. Furthermore, researchers have been known to insufficiently be trained to design and implement studies in minoritized communities, as well as also having the lack of incentive to recruit and retain sufficient numbers of participants. So again, there's a multifactorial facet to this. And the one thing that comes to my mind is also how some experts believe that there's a fear of exploitation based on unethical practices that have been documented in the past. And this may, again, have made minoritized communities distrustful of the biomedical establishment and thus reluctant to participate, as we see today. And the example that comes, there are several other examples, but the example that we'll use today that I think is much more widely known is the Tuskegee syphilis study. So when reporting the results of research that includes racial and ethnic disparities and inequities, it's really important to discuss, at least with, an evidence-based approach, the implications of these findings for addressing structural racism, which you see here, because these discussions actually affect the study, population, disease, or the disorder studied, and the relevant healthcare systems. And within the structural racism component, you can see that there's multifactorials. There's gap in education, there's the gap in housing, there's the mortality rates that get factored into that. So there's many different reasons for why this happens. So what are key takeaways and lessons learned from previous algorithms and cutoffs? And the ones that come to mind are actually the glomerular effect, the glomerular effect filtration rate, GFR, and PFTs. Didn't have my coffee this morning. So in this article, Dr. Fauzi's team evaluates measurement bias in pulse oximetry among a population of hospitalized patients diagnosed with COVID-19. The authors go on to show how measurement bias was associated with differences in delay in recognizing patients' eligibility for COVID-19 treatment. And they do this with racial and ethnic minoritized groups being more affected noticeably. Now, although this is less visible, you have to think about how this is actually similar with, someone's saying my name, so I'll hold off. Jeff Matthews, you're not, okay, there we go. So although this is less visible in terms of how race and ethnicity based cutoffs and pulmonary function tests and estimated glomerular filtration fractions have led to the underestimation of disease severity among patients of racial and ethnic minoritized groups. and thus that has led to delayed treatment of severe lung disease, which has also then led to limited access to timely kidney replacement therapy and transplantation. We should also be thinking about how is it also different? So having spoken about this, one could actually argue that race and ethnicity based discrepancy of pulse oximetry exposes a fundamental flaw in the acquisition rather than interpretation of data. Now, although these biases are, they're all associated with some type of systemic underdiagnoses of different disease processes, as well as withholding of therapies for racial and ethnic minoritized groups, you have to kind of think about this, the similarity there. One is where you acquire data where it inherently is biased towards treating a certain group of people more efficiently versus using a cutoff to delineate how I'm going to use data or how I'm going to use management and treatment to apply to a patient of a certain group or population and based on race. And this is actually, and this is so problematic because of the root of the GFR and the root of PFTs they were all framed from a biological construct. And so that's how these algorithms came into play. So there's so much harm that can be caused when we really don't appropriately address race and ethnicity and what that really that truly means and how it plays within our society. So I want to also get into the New England Journal of Medicine article. I went ahead and copied the most important tables here, which look at the GFR, and it looks at how it factored race in. It looks at how, again, both equations report higher GFR values and how that leads, unfortunately, in Black individuals to suggesting that they have better kidney function and then further delay into treatment and potential kidney transplantation. Same as well with the PFT. So I'm just reiterating here again, you include race based off of a biological construct, and then it leads to inaccurate estimates of lung function, which then results in misclassification of disease severity. So I want to highlight the necessity and the benefit of ensuring DEI in all aspects of work. Why does DEI matter? Well, DEI is the base to build on if we really want to address these health disparities. So DEI allows us to gain perspectives and how to approach a problem. It allows us to make conscious efforts to address bias. It allows us to be truly inclusive. It allows team members to be accountable while we're focusing on being equitable in our projects. So I wanted to use this platform to put in a plug to really think about how we're recruiting patients, what we're really looking at, what we're saying after we've acquired our data, because we really don't want to perpetuate health disparities while having the good intentions of trying to address them. And so it's really important to have a DEI lens as we acquire data, formulate our team, and create a collaborative effort in really trying to mitigate these biases within pulse oximetry. Thank you so much.
Cody, thank you very much for joining us. Appreciate your time. And we will jump into our next speaker. And if you're able to stick around for some discussion that may follow later this morning, that would be great. But we know you also have some classes to teach this morning. So thanks again for taking the time. Next up is going to be James Lee from the FDA. As you're sharing your slides, I'll just do a brief introduction and feel free to elaborate, but the screen is yours. Dr. James Lee is the Director of Division of Sleep, Disorder, Breathing, Respiratory, and Anesthesia at the FDA Center for Devices and Radiological Health. The division regulates medical devices that are common in intensive care units, operating rooms, and to treat and diagnose obstructive sleep apnea. Dr. Lee has graduate degrees in biomedical engineering and material science. Thanks so much for joining us today. The floor is yours.
Thank you, Dr. Can you hear me?
Loud and clear.
Okay, great. Thanks so much. Thank you, Julian. Good morning, everyone. Thank you. I'm here to talk about pulse oximeters and U.S. regulation. And regulation of prescription pulse oximeters are based on a moderate level of device risk, meaning these devices are reviewed under the FDA program for 510Ks and are cleared under the basis of substantial equivalence. As described in our 2013 guidance, document for pulse oximeters, these devices undergo clinical testing, bench testing, and other standardized tests like biocompatibility to review the device's relative safety and effectiveness against predicates with the same intended use. These devices are considered for spot checking, trending tools, and are not generally for diagnosis of a disease. A cornerstone of the FDA guidance and review is the use of the international standards for recognized evaluation of pulse oximeters. And certainly this standard establishes the basic safety needed and defines protocols and tests that are utilized in evaluation of pulse oximeter and its measurement of defined performance. These pulse oximeters are labeled for trending or spot checking for oxygen saturation levels of patients in hospitals, doctors' offices, and also prescribed for home use by patients under the care of a physician. Regarding what is considered to be over-the-counter regulation of these devices are under enforcement discretion and fall under product codes like PGJ and OCH. These are specifically for general wellness, which apply for sports and aviation uses and are not intended for medical purposes. For this reason, they do not undergo FDA pre-market review and can be sold directly to customers. Please see our guidance in the link below, and that's our general wellness guidance defining what's low risk. Following up here, the FDA reviews medical technology like oximetry under established regulations under the Code of Federal Regulations, or CFR. Here we would like to provide some established definitions of these regulations. Furthermore, the FDA buckets or groups within these regulations by product codes, which allow for a more granular definition of products and allow for detailed assignment according to both established regulations and sorting devices by type. What should be noted is that oximeters devices utilize both visible and invisible wavelengths of light that rely on absorption of tissues where the remaining signal can be transmitted to the opposing sensor, like in the fingertip pulse oximeters we review. Innate configurations like this, while effective in picking up physical signals, have limitations due to technology and configurations of the sensor system. Highlighted here are several confounding factors that may contribute to either inaccuracies of the signal or increase the variability in relation to ground truth or absolute oxygen saturation in the hemoglobin. One of the main considerations is the level of skin pigmentation, which is a major part of our discussion today. In addition, there are other patient dispositions that may raise challenges regarding the device accuracy and signal quality, like the ratio of oxygenated and deoxygenated hemoglobin, which does not include consideration for other types of hemoglobin. Therefore, disorders of the hemoglobin molecule or anemia may cause a reduction in pulse oximeter accuracy, in addition to other factors like intravascular dyes, tattoos, nail polish, and ambient light. It may interfere with the sensor and contribute to lower accuracy. For pre-market desaturation studies, as per the FDA 2013 Pulse Ox guidance, in vivo pre-market desaturation tests for SpO2 accuracy is recommended under laboratory conditions for all new pulse oximeters, as well as all previously cleared pulse oximeters that have undergone major electro-optical sensor modification and or algorithm changes to the SpO2. In general, the purpose of in vivo pre-market desaturation studies is to verify the SpO2 accuracy of the pulse oximeter device performance in comparison to the gold standard measurements of blood SaO2 measured by co-oximetry under a specific SaO2 range, generally 70 to 100%. Therefore, typical labeling is for general indication for non-invasive measurements of blood oxygen saturation. If a manufacturer wishes to seek a particular clinical indication for the use of the pulse oximeter, for example, to screen or diagnose a particular disease or condition, then we would recommend additional clinical safety and effectiveness data that would be submitted in review. Regarding the subjects, as you know, per the Annex EE of the ISO 80601-2-62, the pre-market desaturation study should include sufficient number of patients or subjects to attain statistically significant necessary information to demonstrate a specific SpO2 accuracy. Furthermore, a minimum of 200 pooled data pairs, SpO2 or SaO2, distribute evenly over testing range, the aforementioned 70 to 100%, with a minimum of at least 10 healthy volunteers is recommended. As per the FDA guidance, the study should have subjects that range in age, gender, skin pigmentation. FDA guidance recommends at least 2 subjects or 15% of the subjects, whichever is greater, to be darkly pigmented. Per ISO standards, carboxyhemoglobin should be less than 3%, methemoglobin should be less than 2, and the total hemoglobin in grams per deciliter should be at least 10. Per ISO standards, allowable test conditions for pre-market desaturation studies include application of warming techniques to improve circulation, pulse amplitude at the pulse oximeter probe site, covering of the pulse oximeter probes with opaque material to prevent optical interference, and the addition of carbon dioxide gas added to the inspired gas to maintain normocarbia and to prevent alkalosis secondary to hyperventilation caused by hypoxia. A catheter, and during the studies and test procedures, a catheter is placed within the artery of all test subjects prior to desaturation. A cleared pulse oximeter is placed as a reference to detect stable plateaus. The fraction of inspired oxygen varied in a stepwise manner to achieve a series of targeted steady state saturation periods. When the reference pulse oximeter stabilizes for 30 seconds or so, the arterial blood sample is measured and taken and compared simultaneously to data pairs, so that basically you could measure SpO2 and SaO2 variance in real time. Per the FDA guidance for data submission, all following data as it pertains to pre-market desaturation study and testing is requested to be submitted during the FDA review. Pertinent test apparatus used, inclusion-exclusion criteria, the number of samples taken per subject, specific conditions during the test, such as motion, low pulse, amplitude, laboratory conditions, the type and frequency of motion of testing, if applicable, and the criteria and methods to determine the stability of the reference arterial blood oxygen saturation at the pulse oximeter site. Desaturation profile, including target plateaus and ranges, as well as formulae for determining the average root mean squared. So during the analysis of a pulse oximeter that's derived from pooled data pairs, all subjects within the entire test range, typically 70 to 100%, currently clinically relevant thresholds like 94% or 88%, or the range 86 to 94%, let's say, are not analyzed further. Importantly, the study is not particularly powered to determine significant differences between cohorts, such as age, sex, or specific pigmentation values. Regarding the recommendations for the analysis that the agency asks, additionally, individual data pairs as well as pooled data pairs are requested to be plotted in a modified Blant-Altman plot. These plots on the right side of the slide have the SaO2 on the x-axis and the pairs, the differences between the SpO2 and SaO2 on the y-axis. FDA guidance also requests population mean bias between subject variances, within subject variances, as well as the upper 95% and lower 95% limits of agreement. FDA asks certainly for discussion and rationale why any data points would be excluded during a sponsor's analysis and presentation. Regarding outliers, as per the FDA guidance, all noticeable outliers require discussion of the state of health of the subject's characteristics, certainly the test setup, test procedures, and any other factors that may affect these data points. Also, a discussion how outliers do not raise questions of safety and performance or concerns regarding the accuracy of the device is requested as well in our review. So, what are some of the questions about that the agency has about the clinical desaturation studies as they are, currently received? Given the limitation and purposes of pre-market desaturation testing, it is important to consider the following questions. Currently, pulse oximeters are not diagnostic medical devices. Instead, they are verified to be estimates of SpO2 within 2 ARMS of the 95% of the data pairs. In this function, they are tools and they practice interpretation and verification of the SpO2 is particularly relegated to the clinician or the learned intermediary looking at the signal and values. In the current indication, adequate for clinical decision making, these are some of the questions we have. In addition, are some of the clinically relevant ranges or thresholds, whatever the degree of certainty is required, and what is the need for accuracy for critical values and ranges. And the second question would be, can current pre-market desaturation studies be improved upon to provide clinically relevant pulse oximeter performances for all populations and settings? At the end of the talk, we provided here some links, including our guidance for 510Ks, general wellness, and the mention regarding the advisory panel meeting and the accuracy safety statement the agency put out last year. Thank you so much for your time, and look forward to the discussion.
Thank you very much. Please, everyone, feel free to add to the chat questions and comments. We're going to keep moving on in the agenda, though. There will be time for discussion later this morning. I'm going to hand over at this point to Sandy Weininger and Bob Kapadik to help us wrap up this opening session, and then we're going to move on with the program. For everybody who's joining online, we are running about 15 minutes behind in the printed agenda, but we are going to get through everything, so hang in there. And for those of you who are here in person, please don't all leave at once, but if you are dozing off at any point, there is fresh coffee outside, so it is there. Sandy, over to you.
Thank you so much. Good morning. My name is Sandy Weininger with the FDA. I had intended to be there in person, but my back had other ideas about what to do. I am going to give you an overview of standards. My complete presentation I sent to Fifi and she will post it, but I'm just going to try to give you the highlights in the interest of saving some time here. So let me see if I can actually make this work. Well, let's hit this button and see what happens. That's the wrong screen. Swap. All right. Can you see my screen?
Yes, you're all good.
Okay, very good. So we're going to talk a little bit about ISO 80601-2-61, which is our pulse oximeter standard. And I want to tell you a little bit about what it is and how it fits within the big umbrella. Just briefly, we've been looking at standards to cover the safety of pulse oximeters since roughly 1982, 1986 is when the first inklings of the standards came about. And you can read through here about all the different artifacts, devices, standards, whatever you want to call them, that were produced to try to assure safety. 601 or our standard 80601-2-61 fits at the center of a whole family of standards. We have a standard for quality systems, right? Every manufacturer who does design and development has to have an established process for doing so. And it is, at least in the US and most other countries, it's evaluated by some third party to make sure that it is functioning. And we can talk about what functioning means later. There's also a process for identifying hazardous situations. So the standard does a good job of identifying a lot of aspects of things that can go wrong, but there may be things that aren't included in the standard. And this standard, ISO 14971, risk management, helps to try to identify the additional pieces that might need to be identified, reconciled, and have their risks controlled to a reasonable level. And then, so those establish processes that a manufacturer has to incorporate into their design development process. Now, they also have to make good decisions within that design and development process. And that's where this family, whoops, there's timing here, sorry. That's where this family of 60601 comes in. IEC 60601 is the requirements. You can see medical electrical equipment, general requirements are basic safety and essential performance. And so it establishes acceptance criteria. For example, how hot can your probe be, right? And how accurate should your probe be? Now, we also need to talk about the differences between what an engineering standard is and what a clinical perspective or clinical standard might be. The standards that we are dealing with within ISO are device construction standards. You can see over here in the pink, this is what our scope is, right? Is the device safe? And we, for terms of art, define safety as basic safety, safety from physical hazards, and essential performance. Does your device perform in an acceptable way? But importantly, that's asking because I need to test my device and I need to know how bad certain functions will behave and still be acceptable, right? That's not applying it to the patient and saying, is 3 or 4% change in saturation good enough? That is not what our engineering standard's about. It is certainly very important and very germane to today's discussion for us to figure out what we mean by clinically significant differences. If we're talking about pigmentation and I have 10 buckets and those buckets are off by 1/4 or 0.5%, is that clinically significant? That's the challenge that we have to wrestle with today. And how do we identify credible reference devices in order that we can have confidence in the objective scientific evidence brought to bear on this problem? 601 is a pretty broad standard. Not only does it talk about the basic safety and essential performance, but it has what are called collateral standards. And these collateral standards augment the standard. And the reason that they're not in the general standard themselves is because this would be 2,000 pages and no one would be able to lift the standard if everything else was included. But you can see here are some of the collaterals. There's a collateral for electromagnetic compatibility. There's a collateral for usability. There's a collateral for environmental impact, which we frankly don't use. There's a collateral for home use. There's a collateral for alarms. There's a collateral if you're going to use your device in a closed loop physiologic construct. And there's a collateral for emergency medical systems. These add additional, usually more conservative requirements on the robustness of your device, particularly the EMS and the home use. They talk about shock and vibration so that your device actually survives after you drop it on the floor, for example, or if you're bouncing around in the back of an ambulance. And EMC, electromagnetic compatibility, if you bring your cell phone right next to the front end of many of the physiologic monitoring devices, there's enough energy put out from your phone that it completely obliterates any signal you might get. Right? So that has to be handled, and it is handled by this series of standards. And this is just for completeness. This is a list of those names that are in there, and you can peruse this. It'll be posted. And just briefly, the scope of the standard is for 2-wavelength devices that estimate the arterial oxygen hemoglobin saturation and pulse rate. It is not a stand-in for, it is not equivalent to, it is an estimation. And as we have seen, perhaps it is a somewhat flawed, oh, that's not a good word. It is not a perfect estimation of that arterial saturation. So we have to try to communicate that uncertainty to the clinician and to the users of the devices. And in fact, the standard has lots of information that goes into what we call labeling or technical documentation or user manuals. And to the extent that we can augment that, and we have to, we know that they are deficient, that's what we're here today, right? We're here to understand what is the information that we think is essential that we have to augment these standards for. And this thing is going on itself, so that's fine. So I urge you that if you have suggestions, the more concrete and the more specific they are, the easier our jobs will be. So if you have a suggestion, by all means, either type it into the chat or put it into the Google document in the form of a requirement. I think pulse oximeters shall do the following. They shall measure oxygen saturation to within 3%. And, and I shall use 500 subjects to make sure that it does that. Something to that effect. The standard does talk about a minimum uncertainty, and we are using the term accuracy, ARMS, because that is a term that combines both bias and precision into a single metric that can communicate to the user about how accurate their product, or about how different, how off from the objective reference devices your product should be. And we talk about ranges of 70 to 100. Really unethical to start testing oximeters below 70 because those desaturations get to be a little dangerous. But we think that from testing from 70 to 100 is fully acceptable when done with the proper safeguards. We define the formula so that we can all talk about the accuracy of an oximeter using the same definitions. One of the major purposes of standards is to define everything possible, so that there is no uncertainty or confusion about what a particular value means. And we define the test methods for evaluating the effectiveness. And I'll just say one more brief word here. These test methods are designed to demonstrate that your oximeter, when I say your oximeter, a new oximeter that someone wants to place on the market is not out to lunch in terms of its performance. It is not an exhaustive means for demonstrating how to calibrate an oximeter. It is simply a qualification test based on a limited number of samples to make sure that the products you're putting on the market perform up to some minimum standards. You, as a purchaser of a particular device, are certainly free to augment and do more. And in fact, in some cases, the FDA guidance document has additional requirements because we thought that they should be more stringent. Standard talks about what goes into the clinical subject population. It does not do a good job, and that's, again, why we're here today, and we're going to fix that. Concrete requirements that we can put into our standard are requested and very strongly. You can read these. These are also the factors that influence bad performance, right? Everything that shouldn't, that causes an emergent behavior that we don't want, right? When we create systems, we create emergent behaviors. Sometimes we create emergent behaviors that we don't want. To the extent of our knowledge, those should be listed, and manufacturers should identify what they're doing to control the risk that arises from them. For example, delay time. How long should it take for my oximeter to churn through the data before it spits out a particular value? And is that a hazardous situation if it takes too long? And if my oximeter holds values that are 15 minutes old on the front panel, you might think that's not such a good idea. Okay. And I am going to leave it there in the hopes that we can somehow get back on track.
Sandy, thanks so much. We're going to go ahead and move on. There's some great questions. Jim, thanks for the question in the chat. I think there's going to be some discussion on that tomorrow, but we're going to flag that. Special thanks to Dr. Ehi, Dr. Lee, Dr. Weininger for joining the first session, and we're going to roll straight into the next session on real-world challenges in pulse oximetry.